Propagation at Pizzo Native Plant Nursery starts when we collect seed from our 30-acre prairie or from one of our many established seed beds. We have several vendors that we purchase seed from as well if we don't have those species available to us here at the nursery. When the seed is determined to be ready for collection, it is carefully cut, bagged, logged, and placed into proper storage. Seed readiness is observed in many ways. You might hear the seed shake in the pod. The pod might crack open, crumble, or shatter. Seed readiness is both visual and auditory. The seed you see being collected here, Baptisia australis, will turn brown and you can hear the seed shake inside the pod. When collection is completed, the seed is brought inside to be logged and stored. The species code, date, location, and source are recorded in the collection log sheet and written on the collection bag as well. The seed is given a numerical lot number that includes the species code, year collected, and its source. If a customer ever needs to know the ecotype, recorded as the county of original collection, we are able to supply that information. The seed germination code is noted as well to determine what the next steps are for the seed. We keep detailed information about all the lots collected, and we are always mindful of only collecting 20 to 30% of the population for our use. All information is stored in our computer database, where all future actions will also be tracked. We have several partnerships with local park districts and forest preserves for seed collections and trades. Many of our own employees will harvest seed from their own personal gardens. We collect close to 200 lots of seeds each year from our own prairie, Jack Pizzo's prairie, seed beds, and employee gardens. We are always on the lookout for new sources of seed. Depending on its germination code, a collection may go straight to the seed room for dry storage or taken to the seed cage for cleaning. The seed we harvest is for our purposes, so therefore larger industrial-size equipment is not necessary. Once a seed is dried and ready for cleaning, we choose one of several methods. A simple cleaning method is to gently rub the dry seed in a circular motion over the top of a screen into a bin. The ripened seed will fall to the bottom of the bin, and the chaff will remain on top. Not all seeds will come clean, so we collect and save all the chaff in larger bins. In the late fall, after we burn the prairie in seed beds, we will disperse the chaff in troubled areas of the prairie. This is proven to be a beneficial method of restoration. Seed like this Polemonium reptas is self-cleaning and will disperse itself when ready. Makes for very easy cleaning. Seed collection starts as early as May and runs through the end of November. Seeds come in all different shapes, sizes, and colors, and all have individual needs when it comes to cleaning and storing them. There can be anywhere from 70 seeds per ounce to 3.2 million seeds per ounce. Storage time can be as short as 10 days to 2 years in cold stratification. We have learned grasses have a higher rate of germination with at least 2 years of dry storage. Certain species are cleaned and sown as soon as they are collected. We keep detailed records of how a seed was cleaned and stored each year and compare notes from one year to the next. Simply applying a gentle pressure to this Potentilla arguta and gently shaking the seed out is all it takes to clean this species. Most of the seed harvested is kept in our climate-controlled dry storage room. The temperature and humidity combined will total a number of 100. Cleaning seed throughout the winter months is a perfect winter activity and ensures work for our full-time, year-round employees. We have over 1,100 lots of seeds in both dry and cold storage, with almost half of the lots collected here at the nursery. We have customized our germination codes to suit our growing methods, and as we continuously strive to improve our seed cleaning methods and storage of our seed, our germination rates increase every year. Once a seed is ready for stratification, the seed is pulled from the dry storage and mixed with a sand-water mixture. We use silica sand manufactured in Weedron, Illinois, and distilled water to be sure any harmful contaminants are left out. The sand and water are carefully stirred together to make a smooth, sticky sandcastle-making consistency. The mixture is immediately ready for use. Any unused mix can be kept covered for future usage. There are other methods of stratification, such as moist coffee filter or moistened peat moss. Sand and water work best for us as we are able to see the seed through the clear plastic bag it is stored in. The sand mixture is then scooped into the seed bag and gently shaken up with the seed. Care is taken to be sure all the seed has contact with the wet sand. We then flatten the bag for better storage purposes. The bag is then placed in the fridge and kept the assigned number of days, anywhere from 10 days to 120 days before it is ready for sowing. We have seed in storage for over 10 years that is still viable. Proper storage is key to successful germination. We conduct germination tests to determine viability of specific seed lots. The nursery manager will communicate to the nursery propagator through our database which species need to be sown. A sow sheet is composed, labels are printed, and the seeds are sown. Sowing is conducted in either our propagation house or greenhouse depending on the needs of the seed being sown. Trays are filled with germination mix, a blend of fine-grade peat moss, perlite, and vermiculite, certified by Veriflora. The trays are labeled with the specific lot and week being sown. We use 3 different tray sizes: a 1-cell community tray, a 288-cell tray, shown here, and a 432-cell tray. There are several sowing methods used at the nursery. Either by hand or with a vibro hand seeder are favored. If a seed has been in stratification, it will be dried before sowing. Keeping or adding dry silica sand to the seed allows for higher visibility and a more even distribution of seed into the individual cells. If necessary, the sown trays are lightly covered with the germination mix and then set down in the assigned location. There are seeds that do not require any cover at all. The seedling trays are placed in a location that best suits its germination needs and watered immediately after they are set down. Under trays are used on the ground to keep the trays from getting too wet. The tables are reserved for the 432 trays, and we also have ebb and flow tables for wetland species. Once the seed has germination, its germination date is recorded and inventory is done to communicate to the production team the number of seedlings that are ready for transplanting. Genetic diversity is important to native plant propagation. We believe genetic diversity holds the key to adaptability and resilience in native plant propagation. Once the seedlings are transplanted, they are set down in their appropriate location in the nursery hoop houses and covered with rice hulls. Rice hulls are used for both moisture and pest control. This is a brief overview of our propagation department here at Piso Native Plant Nursery.